Hello everyone, Mr. Walker here. This lesson is going to be on mitosis and meiosis. And just before we do get into the details of those two kinds of cell division, just a little bit of a reminder about the cell cycle. So remember that cell division only involves one of these pi wedges within the cell cycle. So I'll put D for division, and the division of the cell includes the processes of, I'll put here M for mitosis, and C4 cytokinesis. We also have these other wedges of the pie here. So going in this direction, this would be G1 or growth 1, gap 1, goes by various different names. This would be S, which stands for synthesis. And over here, this is G2, another kind of growth and preparation for cell division. So starting at the end of the cytokinesis, following this arrow all the way through into the beginning of M. So G1, S, and G2, all of those are referred to as interphase. Interphase is not part of cell division. It is not part of mitosis, but we do have to have something taking place in interphase and specifically during S of interphase of the cell cycle before cell division actually begins. And what that is going to be is that during S, again, S is for synthesis, and what we're talking about is the synthesis of DNA and the duplication of the chromosomes. So once the chromosomes have been duplicated during S of interphase, then the cell can continue on through G2, growing and preparing for cell division. But the actual division of the chromosomes, that is mitosis. Division of everything else within the cell, that is referred to as cytokinesis, and usually there is a little bit of a overlap between the two of them. A little bit of a reminder also of the time that the cell does spend during each of these. On average, for most cells, it is about 90%. That is the number that we typically use. 90% of the time of the cell cycle is spent during interphase, and that leaves somewhere around the remaining 10% when the cell is actually dividing. So again, this lesson is going to really be focusing on cell division. So we'll start off with mitosis and then go on to meiosis. Mitosis is the process whereby we have genetically identical cells that are being produced. Okay, mitosis. Mitosis is also referred to as conservation cell division. So if we do have a human cell, which has 46 chromosomes, we're then going to generate two cells which are going to have exactly the same number of chromosomes, 46 chromosomes, not only the same number, but they will be genetically identical, the same genes and the same alleles, in fact, that you do find on the chromosomes in those two cells. We can also say then that when we are talking about the 46 chromosomes, it is 23 pairs of chromosomes that we do have. So the diploid number would be 2n. And at the end, we get two cells which also have the diploid number. All right, so we're starting with one cell, we're ending up with two cells, we're conserving the chromosome number, and we're generating genetically identical cells. So going back to what is taking place during S of the interphase portion of the cell cycle once again, if we do take a look at the chromosomes, if it is a human cell, remember that within the human cell, there are, well, 23 of the paternal chromosomes, which will be at different lengths. Some of them will be longer, some of them will be shorter. And we have an equal number of the paternal chromosomes as well. So what is going to have to take place? I didn't draw 46 here, but in a human cell, again, there would be 46 of these lines. What has to happen during S of the cell cycle is for every single one of these, we do need to make another copy. And this is going to be an exact copy that we're making for all of the paternal chromosomes that I'm showing in blue and the maternal chromosomes that I'm showing in red as well. Okay, so that is what's going to be taking place during interphase. Now remember that when the chromosomes are in interphase, they're in this long and stringy form that we refer to as chromatin. So if this is our cell, and if I draw the nucleus within the cell. Here I'm just going to focus on one pair of chromosomes, but exactly the same process would be taking place with the other 44 chromosomes if it's a human cell, or the other 22 pairs of chromosomes. So the DNA is in the form of the long and stringy chromatin. And for this paternal chromosome, what has to happen during S of the cell cycle is the duplication of it. Now remember, when we do duplicate the chromosomes, they are held together by the centromere. These two chromosomes, even though we have duplicated the number because they are held together, we still do refer to them as a single chromosome. So even though we do have this duplication of the paternal and the maternal chromosome, we are still going to call this cell a diploid cell, two copies of each one of the chromosomes. As a reminder as well, when we do talk about these individual halves of the duplicated chromosome, they are now referred to as the chromatids, ones that are attached by the same centromere. So these two here would be called sister chromatids. These two here would be called sister chromatids. And if we compare this blue one to the other homologue of this pair, we would call these the non-sister chromatids. So all of this again is going to be taking place during interphase of the cell cycle. So now as we go on to mitosis, so we transition through S to G2 and then into M of the cell cycle, we're going to see a few changes that are taking place. A couple of key things is the nuclear membrane is going to disappear. So now the duplicated chromosomes, they are free to move around anywhere within the cell and in no particular orientation. A couple of other things that are going to be forming, we're going to have from what are referred to as centrosomes, two different centrioles. And usually they show them as two cylinders kind of at right angles to each other. So we're going to have two of these centrosomes, and they're going to migrate to opposite ends of the cell, which is what we refer to as the poles. So I'll draw this kind of like a globe, where up here at the top, we would have one of the poles, and down at the bottom, we would have the other one. So those are our two poles, and that is where these centrioles are going to migrate to. So what is really important is that coming out of these centrioles are going to be microtubules that make up what are called spindle fibers. So these spindle fibers are going to be what move the chromosomes around within the cell. So they are going to, as I've shown with this one here, attach to the centromeres on each one of the duplicated chromosomes, and that's what's going to enable them to be moved around. So at this point, the duplicated chromosomes, they can be found once again anywhere within the cell. We have these centrioles that have formed and migrated toward the poles. We have the spindle fibers that are going to be forming. Our nuclear membrane has disappeared. And now what we're also going to be able to see is the chromosomes. So the chromosomes are going to go from the chromatin form to the condensed chromosome form. So now instead of a long stringy chromosome, we're going to have one that is thicker, that's wrapped around the histone proteins. And we usually show this as just, well, having these thicker lines like this, kind of forming an X-shaped structure, but this is not an X chromosome that we are taking a look at here. I'll do the same thing with the maternal chromosome. So it's kind of the same thing. What it's showing is that we do have two sides. They are still held together by the centromere in the middle. Ones that are held together, again, those are going to be the sister chromatids. Now we can talk about these two collectively as one of two things. We can call them homologous chromosomes because they are a pair, they are of the same chromosome number. And we can also refer to this as a tetrad because we have one, two, three, four copies of this particular set of genetic information. These particular genes that we find on these chromosomes, on this chromosome number. So this structure that I've drawn here may not be entirely accurate. So when we do have duplicated chromosomes and they're condensed, they're probably more likely sandwiched together, held together by the centromeres here, and sort of the same thing that we see with the paternal chromosome, more sandwiched together. Just to make it a little bit easier for me to draw these, I'm going to continue to draw them just as a single line, only now when I draw these single lines, they will be in fact referring to the condensed chromosomes. I'm only going to draw the one pair of chromosomes, but once again, this is going to be the case with all other 23 pairs of chromosomes. So we will have our duplicated paternal chromosome, again, that I'll show in the blue. We'll have our duplicated maternal chromosome that I'll show in the red. So what I mentioned so far, as the cell does go into prophase, are really three key events that you should keep in mind. So during prophase of mitosis, these are key events that are going to be taking place. The nuclear membrane is going to disappear. The centrioles and the spindle fibers, they are going to form, and the chromosomes are going to condense and they are going to become visible. So those are key features that you should remember with the process of prophase. Each one of these different phases of mitosis, they're just going to automatically transition into the next one. And the next phase after prophase is what is called metaphase. During metaphase, the spindle fibers that are originating from the centrioles that are now at the poles, they're going to move the chromosomes around within the cell. And where they're going to move them to is along the middle of the cell. So here, the middle of the cell, I'll draw going across this way. And the middle of the cell is referred to as one of two things, either the equatorial plate. Again, if you think in terms of the globe, we have the two poles, and in between we have the equator. So this is the equatorial plate, or it's also referred to as the metaphase plate. So what happens here is those spindle fibers, they are going to be attaching to the centromere of each one of the chromosomes, and they're going to move that centromere so it's right along the equatorial plate. This once again showing the two chromosomes that I drew before. So over here, we might have the paternal chromosome, and lined up in a straight line, all of the chromosomes, at least the centromeres, will be lined up on a straight line. The arms of the chromosomes, they might go up or down in either direction, but they'll be lined up along the center of the cell. So this is the characteristic feature that you can expect to see when the cell is in metaphase of mitosis. So once again, prophase, nuclear membrane disappears, chromosomes become visible. Centrioles form the spindle fibers. Metaphase, those spindle fibers are going to arrange the chromosomes along the equatorial plate. So now those spindle fibers, they're going to contract and they're going to pull the chromatids in opposite directions. So the spindle fibers attaching to the centromeres, and it's going to pull half of the duplicated chromosome in one direction and the other half of the duplicated chromosome in the opposite direction. So it's going to look something like this. From the equatorial plate, we would have one half of the duplicated paternal chromosome, which is now referred to as a chromosome in itself, going in one direction, and the other one that's going in the opposite direction. And again, the spindle fibers is what is going to be pulling those chromosomes in the opposite direction. Again, they're no longer referred to as chromatids. As soon as they are pulled apart, they are chromosomes once again. Same thing with the paternal chromosome. Spindle fibers are going to attach to those and pull them in the opposite direction. So the same thing is going to happen with all of the other 23 pairs of chromosomes. So this process that we're seeing now, where the chromosomes are being pulled apart, this is now referred to as anaphase. During anaphase, those chromosomes that were lined up along the equatorial plate, duplicated portions, the chromatids are pulled apart. They're moving toward the poles, and as they are moving toward the poles, that is now anaphase. Once we do have the chromosomes that have, well, pretty much reached the poles, now we go into the final stage. So now they're at the poles here, the maternal and the paternal chromosomes. We're going to have the nuclear membrane that's going to start to reform around the chromosomes that are now at the poles. And we're going to have the chromosomes that are going to decondense. So it's really kind of a reverse of what we saw with prophase. Again, prophase, nuclear membranes became visible, or nuclear membrane disappeared, and here it's going to reappear. The chromosomes became visible in prophase. Now they're going to disappear and return to that chromatin form once they're going into the last one, which I'll write way down at the bottom here, and that is telophase. So in terms of the different phases that we do have, this is the order. So it is prophase, followed by metaphase, and then anaphase, and then finally telophase. What about that process of cytokinesis? Well, in addition to the chromosomes moving around, we're also going to have the cytoplasmic component kind of evenly divvied up between the two cells. And eventually what is going to happen, at least with an animal cell, is it's going to pinch in the middle. And then eventually that's just going to continue, and we end up having two cells with where we started with one. So just as a reminder, these two cells that we do get, they are genetically identical to each other. We have conserved the chromosome number. We duplicated the chromosomes once, and we divided the chromosomes up once. Where this takes place is wherever we need to produce genetically identical cells. So when we go from the zygote through the embryo and the fetus, all of these cell divisions that are taking place during the growth of the embryo and the fetus, that's going to involve the process of mitosis. In an adult body where we're continually regenerating and replacing cells, these are genetically identical cells, and that is going to be the process of mitosis as well that is involved. So what about the other kind of cell division? So the other kind of cell division is going to be a reduction cell division. So now we're going to start with our two copies of each chromosome in those cells, and we're going to generate cells that are now going to have the haploid number of chromosomes, only one copy of each chromosome. So we're going from diploid to haploid. Here, just like with mitosis, we're going to have to duplicate the chromosomes before we have the division. And once again, it's going to be one duplication of the chromosomes. It doesn't matter what kind of cell division it is, mitosis or meiosis, we're going to duplicate the chromosomes only once. With mitosis, we divided the chromosomes up once. Here, we're going to divide the chromosomes twice. So what that ends up doing, though, is we start with one cell, and we don't just generate two cells, but we generate four cells. What we're also going to see here is that the cells that are eventually produced are not going to be genetically identical cells. They're going to be different. And in fact, this is really important that they're going to be different, because the process of meiosis is the process that does generate the gametes. So again, when we're talking about the gametes, that is the sperm and that is the eggs. No two sperm, no two eggs are going to be genetically identical. And this process of meiosis is going to ensure that there is variation and that no two sperm or no two eggs do end up being genetically identical. So if I draw my original cell here, so this would be a cell inside of the testes or the ovaries, because that, of course, is where the process of spermatogenesis in males and oogenesis in females is going to be taking place. Starts off very similar to what we saw with mitosis. We have the nuclear membrane. Here I'm going to have to draw two pairs of chromosomes. That's the minimum that I would have to draw in order to show what is taking place in the process of meiosis. So we'll start off with a cell that is in interphase, and I'm going to draw two paternal chromosomes, one a little bit longer than the other one, and I'll draw two of the maternal chromosomes, again, one a little bit longer than the other one. So the two on the left-hand side are longer. We will say that they are of the same chromosome number. The two on the right, a little bit shorter, they are of the same chromosome number as well. So before anything else happens, again, we can't actually see this because they're in the form of the chromatin, but we are going to duplicate each one of these chromosomes, the maternal and the maternal paternal chromosomes. So that during S of the cell cycle, only now it is meiosis. So we are going to go on to a prophase, but in this case here with meiosis, we're going to have two different prophase, two different metaphase, anaphase, and telophase. So we're going to go through what is referred to as prophase 1, and then we're going to go into metaphase 1, anaphase 1, telophase 1, and then we're going to go through these phases again, only they're going to be prophase, metaphase, anaphase, and telophase 2, Roman numeral 2. Okay, so we're going to go through each of these twice. So what goes on in prophase 1? In some ways, it's similar to what we saw with mitosis. So we're going to have, once again, the nuclear membrane that disappears. We're going to have the chromosomes that condense. I'm not going to show them any different here. We're just going to assume that they are going to be condensed chromosomes. We're going to have, once again, those centrioles that are going to form, because we're still going to need those spindle fibers that are going to be moving the chromosomes around in the cell. But here we're going to have something else that's taking place as well. What's going to happen with the homologous chromosomes is they're going to come into close contact with each other. Again, this can be anywhere within the cell that this is taking place because there is no nucleus and the chromosomes are free to go anywhere within the cell. So what we're talking about now is prophase, and this is a distinct difference that we're going to see between prophase 1 of meiosis and the prophase that we saw with mitosis. And the event that's going to take place here is referred to as synapsis and crossing over. Synapsis and crossing over. This is really, really important. So for the synapsis, I'm just going to redraw these two chromosomes here. What we're going to have is the arms of the chromatids are going to come into close proximity. And here we're talking about the arms that are going to be the non-sister chromatids. They're going to come into close proximity, and that is referred to as the synapsis. And what we're going to have going on is this crossover event. So where I have these two segments that are overlapping between the non-sister chromatids, we're going to exchange the information. So when we do exchange that information, what we're going to end up with is some new chromosomes or chromatids that don't look entirely like the original ones, which were entirely red in color or entirely blue in color. So the same thing is going to be taking place with all of the chromosomes. I'm just going to show it on this one. So when we do have this crossing over that does take place, I'll redraw these again. So now we're going to have the paternal chromosome. This one I'm just going to show as still being entirely red, but the other one I'm going to show it as being mostly red, but a little bit that's blue. So that was the part that was exchanged with the paternal chromosome. So again, I'll keep one of them entirely blue, and the other one is going to have a little bit of the red attached to it. So what I'm now creating here, really important because we want to have variation when we do have the sperm and the eggs that are eventually produced. This is one of the ways that we can get that variation. And again, this is referred to as crossing over. The crossing over has to take place between the non-sister chromatids. Remember that the non-sister chromatids, they have the same genes, they have the same gene arrangement, but they don't necessarily have the same versions of those genes or the same allele. So it has to be crossing over between the non-sister chromatids. So what I've shown here with this pair of chromosomes is going to take place with all of the chromosomes as well. So that is taking place again, and really important that you do remember that that is the source or one source of variation, and that takes place during prophase 1 of meiosis. So now I'm just going to move these chromosomes around a little bit, erase some of the information that we have here. Just like with mitosis, now we're going to shift the chromosomes around, and we're going to shift them along to the middle of the cell. So again, they're going to form along the equatorial plate or the metaphase plate during metaphase 1. But notice that this one is going to be a little bit different. So I'm going to put the longer paternal chromosome here and the other one, which resulted from the crossing over. And when I draw the paternal one, notice that when they're lined up along the equatorial plate, they're lined up a little bit different. So here, they're not actually in a straight line, but in fact, they're arranged in pairs. So this is how you can tell that it's not mitosis, that it is meiosis, is that the chromosomes, they're arranged in pairs. So this is what you would see only one time, and that is during metaphase 1. So now what we would have is the spindle fibers that are going to attach to the centromere, and they're not going to pull the chromatids apart. They're going to pull the entire duplicated chromosomes going in opposite directions. So again, they're going to attach to the centromere, and they're going to pull them toward the poles. So of the tetrad that we have, of the duplicated chromosomes, of the pair that we have that's lining up along the equatorial plate, one of the duplicated chromosomes is going to go in one direction, the other one is going to go in the opposite direction. So I want you to also pay careful attention to the alignment that I have of these chromosomes. So the two that I have at the top, we'll say that they are mostly red. In other words, they're mostly the maternal chromosomes. The ones that I have at the bottom, they are mostly blue. So they are mostly made up of the original paternal chromosomes. In the end, again, we do want to have some variability, and this is the second way that we can get variability. This particular arrangement that I have here is one possible arrangement. So here again, I'm only showing 2 pairs of chromosomes. In reality, we would have 23 pairs of chromosomes in a human cell. So what do I mean that by this is only one of the possible arrangements? Well, what I could also do is just because I do have the one that's mostly red for the longer chromosome that's at the top, that doesn't mean I have to have the one that's mostly red at the top for the other chromosome, or in fact, all of the other chromosome pairs. So in fact, what I could have done is flip these around, and I could have had the mostly blue one at the top and the mostly red one at the bottom. So what this is referred to is independent assortment. Independent assortment. So kind of think of it this way. These chromosomes, they are going to assort or they're going to separate. They're going to move in opposite directions. They're independent of each other though. Just because we have the mostly red one that's at the top for the first longer chromosome, all of the other chromosomes, including the other one that I drew here, they are arranging themselves in pairs along the equatorial or metaphase plate, completely uninfluenced by the position of the original chromosome. So yes, for the longer one, it's mostly red at the top, but that doesn't mean we need to have the one that's mostly red at the top for the other chromosome pair. They are all lining up independent and eventually pulling apart, sorting independent or uninfluenced by all of the other chromosomes. Well, that is going to be our second major source of variation that we do have in terms of meiosis. So this on top of the crossing over pretty much ensures that no two gametes at the end are going to end up with the same alleles. So again, generating unique cells at the end when we finish meiosis. So to continue on with the remainder of meiosis 1, we're still in meiosis 1 here. So we're just going to move the chromosomes, the entire chromosomes, in opposite directions toward the opposite poles. So they were lined up in pairs. The entire chromosome is going to go either toward the top or toward the bottom. So as soon as they start to pull apart at the equatorial plate, then we're going to have anaphase. So that is anaphase 1. This is where the process of segregation is taking place. So independent assortment arranges the chromosomes in pairs in no particular order. Segregation now means that they're pulling apart and going in opposite directions. So just like with mitosis, though, when they do reach the poles, that is now referred to as telophase. So I would then have at the top here, the long one that's mostly red with a little bit of blue, and I might have the shorter one that's mostly blue with a little bit of red. So that means at the other end, I would have to have the shorter one that is mostly red with a little bit of blue, and the longer one that is mostly blue with a little bit of red. So now this is our telophase. So during telophase, we might see the chromosomes decondensing. We might see the nuclear membrane reforming. We would see the separation of all of the other cellular components. So now what we have ended up with is two cells that we do have. Now notice that the original cell did have four lines, right? The one that we started with had a long blue one and a short blue one, and it had a long red one and a short red one. In other words, there were four chromosomes. There were two copies of each, so that was a diploid cell. Now let's take a look at the cells that we have here. As long as they're held together by the centromere, they're still considered one chromosome. So in each of these cells, I don't have four, I only have two, which means what I have already done is I have taken a diploid cell and I produced two haploid cells. Already after meiosis 1, I do have haploid cells. We're not done. We need to continue on with the remainder of meiosis. So we have two cells. We don't duplicate the chromosomes again. That only happens once, and that is prior to meiosis 1. As we go into prophase 2, not that much exciting is happening. Nuclear membrane is going to disappear. Chromosomes are going to condense. Spindle fibers are going to form. Usually the spindle fibers, they're going to form at different orientations. So if here they were at the top and the bottom, with the next division, they would be at the sides. And so in this case, the equator is going to be going vertically up and down. So it's from these centrioles where we're going to have these spindle fibers that are going to be forming. They're going to move the chromosomes around and they're going to line them up along the equatorial plate. So now this is going to look kind of like what we saw with metaphase of mitosis, only what has happened, and I can't remember which chromosomes I had at the top and which ones were at the bottom, but now we're going to line them up along the middle of the cell, only, well, we can tell the crossing over has taken place. So let's say that I have the longer one, and now I have the shorter one, which is the our maternal one with a little bit of the blue, which means that down at the bottom, I would have the shorter one that's mostly blue and a little bit of red, and the other one that's mostly red with a little bit of blue. So how is this different than what we saw with meiosis 1? Well, they're not arranged in pairs. How is it different than mitosis? We can see the crossing over has taken place. But now it's going to be similar in that we're going to have the spindle fibers coming out from the poles, from the centrioles. They're going to attach to the centromeres. And now what they're going to do is they're going to pull the chromatids apart. In mitosis, the chromatids were pulled apart right away. In meiosis, the chromatids are not pulled apart in meiosis 1. That doesn't happen until metaphase 2 of meiosis. So as they are pulled apart, each one of these, of course, are no longer going to be called a chromosome. They will be called a chromatid. So just to make it a little bit easier for myself, if I just draw a line down the middle, the ones on the right-hand side will go toward the right-hand pole in both of these cells, and the ones on the left will go toward the left-hand pole. So if I erase the spindle fibers, and just to simplify it a little bit, I'll erase the centrioles as well, just to see what eventually we do get at the end here in terms of the cells that we do have. So if I just redraw chromosomes that I have in the center, shift them over to the poles. So we're going to have this long blue one here that ends up at the left-hand side. We're going to have the short blue one in the other cell that ends up at the left-hand side. We're going to have the one on the left-hand side that is the short red one. Okay, short red one over here. And we're going to have the long red one at the bottom. Cells on the right-hand side, we're going to have the longer chromosome that has a little bit of the blue, the blue with a little bit of the red. We're going to have the one that's mostly red, the shorter chromosome with a little bit of blue. And the bottom cell, we're going to have the shorter one with a little bit of red and mostly blue, and the other one that's a little bit of blue and mostly red. So when they do reach the pole, that is going to be telophase 2. We no longer have these chromosomes in the middle. We can get rid of those. These chromosomes are going to decondense. Nuclear membrane is going to reform. Cytokinesis is going to take place. And now we have our 4 cells. So if we do take a look at these 4 cells, let's do a couple of things. Well, first of all, we have 4 cells. We started with 1, we end up with 4. If we count the chromosomes in each one of these, there are 2. We started with a cell that had 4 chromosomes. Now we have cells that have 2 chromosomes. This is reduction division. We've taken diploid cells and converted them into haploid cells. What can we see with each one of these cells? Let's take a close look at the chromosomes. Show that, in fact, yes, they are different. So this one at the upper left-hand side, it has a long, entirely blue chromosome and a short, entirely red chromosome. If you take a look at all of the other ones, there are no 2 cells out of these 4 that actually have the same color for those chromosomes. So that is due to 2 things. Why did we get all of this variation? With meiosis, spermatogenesis, oogenesis, the formation of the sperm, the formation of the eggs, there are 2 kind of key things. One is reduction division. We need to take the chromosome number and cut it in half. So if we start with cells that have 46 chromosomes, like human cells, we end up with the sperm and the eggs, the gametes, that have 23 chromosomes. The second key feature that we want to have is variation. How did we get variation? Crossing over. That took place during prophase 1 and independent assortment. And that is taking place during metaphase 1 and kind of transitioning into anaphase 1 as well. Okay, this is how we do get the variation. Just kind of keep in mind as well that there is another source of variation, in fact, the ultimate source of variation, and that is mutations. Mutations to the DNA itself, changing the bases or the letters, as we say, in the DNA. Okay, so we have reduction division. So why is this so important? Well, when we do form the individual in the next generation, what we're going to do is take a cell that has 23 chromosomes, the sperm. We're going to take another cell that has 23 chromosomes, the egg. And as we already know, these 2 are now going to come together. So when they do join together, what do we have? 23 from the sperm, 23 from the egg. This is why we have to have reduction division, so the individual in the next generation, the very first cell, the zygote, and all of the cells that arise from that are going to have the same number of chromosomes as what we saw in every cell from that individual or those individuals in the previous generation. So if we didn't have this process with each successive generation, we would have a doubling in the number of chromosomes.